That means inhibitor. That means materials which stops or which helps in decreasing the reactions are called as inhibitors, or which inhibitors are the materials which resists or which stops or which uh, um, shows their effect in decreasing the activity, or called as inhibitors. And in this enzyme activity also, there are some inhibitors which helps in decreasing the rate of reaction, or which stops the chemical activity of the enzymes. Then we can include inhibitors as a factor like temperature, pH, and substrate concentration. That means this also be treated as a factor, and it is discussed as a separate part. This the inhibitors are discussed as a separate part, and there are three ways. One is competitive inhibitors or competitive inhibition. Then second, that is what is that, non-competitive inhibitors, and the third one is allosteric inhibitors. First, we'll discuss about the competitive inhibitors. This competitive inhibitors, that means here competition between the substrate and inhibitor molecules. Competition between these two molecules, and this will be explained by showing this diagram. Here E means enzyme, and this enzyme is having active sites. Here these are the active sites, and here this molecule is substrate molecule, and this one is the inhibitor molecule. Both are ready to take the place on this active sites. That means the substrate molecule goes and enter into this active site, and the inhibitor takes the site of this one. So 50% of the enzyme is, that means 50% of the active sites are occupied by substrate molecules. By that chemical reactions takes place in their way, and 50% of the active sites are occupied by inhibitors. Inhibitors, they stops the reaction or they stops the activity. So only 50% of the active sites are binded with the substrate molecules, and they forwards the reaction. That means they fit into the active sites of the enzyme, and here 50% of the active sites are fed by the substrate molecules and 50% by the inhibitors. And now this substrate molecules with enzymes, that means a substrate molecule with the support of this active site and the enzyme, they gives enzyme substrate complex. That means both together they gives the enzyme substrate complex, which is a temporary state. As we know that, why? Because we have discussed so many times that by adding this substrate molecule with the active site of the enzyme, they temporarily gives a complex which is called as enzyme substrate complex. And here also in this competitive inhibition, this inhibitors occupies 50% of the active sites of the enzyme, and substrate molecules, they too occupies 50% of the active sites of the enzyme. By this, two complexes are framed. This is a chance of two substrate complex formation. One is this enzyme substrate complex, which is a temporary state. That means a transition state, which is temporary. And this temporary enzyme substrate complex furtherly that dissociates into product and enzyme. That means 50% of active sites are producing end products with enzyme. And what about the inhibitors? They too are occupying 50% of the active sites of the enzyme. And here also a complex will be developed. That is enzyme inhibitor complex. Enzyme inhibitor complex. And this 50% of are occupied by inhibitors, and they blocks the active sites of the enzyme. And as a result of this blocking, products are not developed from this. And as a result of this inhibitors, total production of the production rate will be decreased or reduced. So here this 50% products are reduced by the occupation of inhibitors in the active sites of the enzyme. So this is the competitive inhibitors nature. That means in other activities, what we are observing is active sites are usually they are fed by the substrate molecules, and they togetherly makes a complex which is called as enzyme substrate complex. And this complex state is temporary. Furtherly, that dissociates into product and enzymes as it is. But here, the active sites of the enzymes are occupied by two types of molecules. One is substrate molecule, the other one is inhibitor. Substrate molecules, they occupy 50% of the active sites of the enzyme, and inhibitors, they too occupy 50% of the active sites of the enzyme. As a result of this type of occupation, 50% of the production rate will be decreased or reduced. Then here we are using competitive inhibitors. Yes, this is normal. Then by using competitive inhibitors, how will be the rate of a reaction? Inhibitors compete with the substrate to bind with the active sites. So here inhibitors, they compete with the substrate molecules to bind with the active sites. And here two examples are there. One is succinate that changes into fumarate. Succinate changes into fumarate. In the presence of one enzyme that is called as succinate dehydrogenase. So in the presence of enzyme succinate dehydrogenase, succinate turns into fumarate. But the same reaction in the presence of one inhibitor, that is malonate. This malonate acts as an inhibitor in this reaction. So in the presence of this malonate, succinate, that means succinate molecules are there and malonate molecules are there. So as we said in the first slide, this inhibitors, that means here malonate is acting as an inhibitor. So 50% of the active sites are occupied by malonate inhibitors, and the 50% molecules or substrate molecules are occupies the active sites of the enzyme. So if malonate is added, the 50% of the sites of the enzyme would be occupied by malonate inhibitors, and 50% active sites are occupied by substrate molecule. So how much product formation would be there by this? Means in the first step, 50% of molecules, 50% of substrate molecules turns into end product. In the second step, this 50% decreases into 25, and in the other steps, gradually rate of production of end products gradually they decreases. So here what we can understand, what we have to understand is, by the presence of these inhibitors, gradually the rate of reaction decreases, or gradually production of end products decreases. So this is the competitive inhibitors. And the second example, for this competitive inhibitors is use of sulfur drugs in treatment of bacterial infections. Use of sulfur drugs in treatment of bacterial infections. For this equation, the normal reaction is para-amino-benzoic acid, which is treated as PABA, para-amino-benzoic acid, that turns into folic acid. And this folic acid is required for normal growth and reproduction of bacteria. Usually this is utilized by the microorganisms for their growth, vegetative growth and reproduction. And this is normal reaction. And this PABA changes into folic acid in the presence of an enzyme, that is folic acid synthetase. So here a useful enzyme is folic acid synthetase. In the presence of this folic acid synthetase, PABA turns into folic acid. Now, by using here, sulfur drugs are used as inhibitors. Sulfur drugs are used as inhibitors. So they are going to compete with the PABA and binds with folic acid synthetase enzyme. So 50% of the sulfur molecules, 50% of sulfur and 50% with the PABA, 50% site of 50% sites of the sulfur, this inhibitors occupies active sites of the enzyme, and the PABA occupies 50%. It means the folic acid synthetase suddenly falls down into 50% and gradually decreases. And in very less amount, the synthesis of folic acid. So at this time, bacteria will not able to grow and reproduce. Here, 50% of the sites with sulfur and 50% with PABA. These two molecules, they occupy the folic acid synthetase enzyme active sites, and the folic acid synthesis gradually falls down into 50%, as we discussed it in the first slide. And the rate of production of end products gradually decreases. And in very less amount, synthesis of folic acid takes place gradually. That means production of folic acid decreases. At this time, bacteria will not able to grow and reproduce. That means here substrate molecules, they are gradually decreasing. That means folic acid synthesis is gradually decreasing, and in less percentage of this folic acid, microbes will not grow very well, and they cannot reproduce in a normal rate. So by this, infections will be certain. And here, competitive inhibition is reversible. Why it is reversible? That means by lowering this rate of production of end products, if the substrate, it is a normal way. But if the substrate molecules are more than the inhibitors, then the substrate molecules, which helps in increasing the microbes' growth, are there than the inhibitors. So by this, what happens? This folic acid that encourages the development of microbes. So this is, that's why it is called as the competitive inhibition. Competitive inhibition is reversible mechanism by substrate concentration increase. If the substrate concentration increases, if the folic acid increases, then that helps in the growth of the microbes. So that's why it is called as competitive inhibition, a reversible competitive inhibition. The next, second type of inhibition is non-competitive inhibitors. Non-competitive, that means there is no competition between the substrate molecules and inhibitors. As the name tells, there is no competition between the substrate molecules and the inhibitors. We can take the diagram to understand this. Here, enzyme, it is there, and substrate molecules are there. Take enzyme with active sites and substrate molecules. Here, substrate molecules fit into the enzyme active sites, and the inhibitors does not compete with the substrate molecules. Its structure is completely different. That means here enzyme active sites and substrate molecules. Take enzyme with the active sites and substrate molecules. Here, substrate molecules fit into the enzyme active sites, and inhibitors does not compete with the substrate molecules. That means this inhibitor structure is completely different. That means here we have to recall one theory, that is lock and key mechanism. This inhibitors cannot fit into the active site. That means the shape of the inhibitor is different from the active site. That's why they cannot fit in the active site of the enzyme. So substrate molecules can only fit into the active sites, and they make a bond with the enzyme and gives enzyme substrate complex, which is temporary. And here inhibitors, they cannot fit in the active site of the enzyme, but they can occupy. Just a minute. We can say that inhibitors cannot compete with the substrate molecules. That means they don't compete the substrate molecules to bind with the active sites. So where inhibitor is going to bind means this. That means here is a question where the inhibitors will occupy the place on this enzyme. They are not similar in the shape of that active sites. That's why they are not going towards the active sites, and they are not occupying the active sites of the enzyme. Why? Because of the shape difference. Then where this inhibitors goes and binds with the enzyme. That means this inhibitors binds with the enzyme at the site other than the active sites. That means they are not going towards the active sites. They are taking the place on the enzyme other than the active sites. That means they at anywhere of the enzymes without competition. Anywhere of the enzymes without competition. So here inhibitors, they take the place at anywhere of the enzyme other than the active sites. But after binding with the inhibitors to the enzymes undergoes conformational changes. Yes, in lock and key theory, induced fit theory, each and every enzyme is having two sites. One is catalytic site and the other is buttressing site. So this catalyzing site, that shows conformational changes, and this catalytic site helps in breaking or making of the bonds between the substrate molecules. So as per that, this after binding with the inhibitor, that means the binding of inhibitor with the enzymes results in the conformational changes in the enzyme. So shape of the enzyme changes, even the active sites are changed. That means not only the shape of the enzyme, active sites also be changes their shape, but substrate molecules do not change their shape. They remains as it is. So by this, molecules, they cannot fit into the, they cannot bind with the enzyme active sites. Because of this changed active sites, no product formation takes place. So here, that means first of all, substrate molecules and inhibitor molecules, both are there. This substrate molecule, they occupies the active sites of the enzyme, but inhibitors, they cannot take the position on the enzyme because of their irregular shapes, which do not resemble the shape of the active sites of the enzyme. By this reason, this inhibitors, they occupies the other sites of the enzyme. That means they cannot occupy the active sites of the enzyme. Other than the active sites, this inhibitors goes and binds the enzyme, binds with the enzyme. So after binding with the inhibitors, this enzyme that shows conformational changes. By this, it changes its shape along with the active sites. But here, substrate molecules remains as it is only. That means they cannot, they do not show any change in their shape. As a result of that, there is no bonding between the substrate molecules and this enzyme active sites. So no complex formation takes place and no end products are produced in this type of inhibition. So here in this type of inhibitors, the total reaction, activity will be stopped. That means no end products formation takes place in non-competitive inhibitors. For example, here cyanide binds to cytochrome oxidase. Here, this cytochrome oxidase plays an important role in cellular respiration. Cellular respiration, we know that if the respiration takes within the cell level, that is called cellular respiration. And in this during the cellular respiration, the cytochrome oxidase plays an important role. So here, this is normal reaction. That means cyanide binds to cytochrome oxidase. Then here, non-competitive inhibitor, that is penicillin. Here, penicillin is non-competitive inhibitor of the enzyme required for cell wall formation in bacteria. So this is a irreversible. Just now only we discussed it in a reversible. Competitive inhibitors, they are treated as a reversible reaction. Why? Because as the substrate concentration increases, this reaction again comes back. So it is reversible. But in non-competitive, it is not reversible. It is irreversible. Means that never comes back in giving the starting products or starting material, substrate molecules. So, and this is about the non-competitive inhibitors. So in non-competitive inhibitors, the reaction will be gradually decreased or sometimes completely it will be stopped. That means no end products formation takes place in this non-competitive inhibitors. Then how it happens means enzyme is there with active sites and substrate molecules are there with their own shapes, and inhibitors are also there with their own shapes. But these inhibitors, they cannot take the place of active sites of enzymes. Why? Because of their irregular shape. So they occupies the place on the enzyme other than the active sites. And binding with this binding of inhibitors with the enzyme results in the conformational change of the enzymes along with the active sites. As a result of this, substrate molecules cannot bind with the active sites of the enzyme because of its changed shape. As a result of that, active sites all are free without any substrate molecules and without any inhibitors. As a result of that, there is no complex formation and there is no end product formation. That means totally the reaction will be stopped by this non-competitive inhibitors. So this is about the non-competitive inhibitors. The last and final inhibitors are allosteric inhibitors. These are also called as feedback mechanism. And this product regulates its own formation. That means product formation regulates itself. That means formation of end products, they regulate themselves. That's why it is feedback mechanism. It is called as feedback mechanism. Let us take one example to understand this allosteric inhibitors. Example is glucose. And this glucose, by adding with or by utilizing one ATP molecule in the presence of enzyme hexokinase, turns into glucose 6-phosphate plus ADP. Here, this is the step involved in the respiration, aerobic respiration. First step of aerobic respiration. Here, glucose molecule, by availing one ATP molecule, that turns into glucose 6-phosphate plus ADP. Here, enzyme is hexokinase. And to understand this allosteric inhibitors, or why it is called as feedback mechanism, means let us take this thing. What is that? If the number of end product molecules, suppose in this reaction, this end products should come out number 10. 10 molecules of glucose 6-phosphate should be released during this reaction. But unfortunately, they came only 9. That means one molecule is deficient as per our calculations. So here, number of product molecules are decreased. Then with this low concentration of product molecules, they stimulate the enzyme. They stimulate the enzyme to increase its activity. Then automatically the number of end products molecules increases. That is one situation. In another situation, if the molecules, they should be produced 10 number, but they are produced more than 10 molecules. That is also a imbalancing character. Then how it will be regulated? If they are produced less than the expectation, then they stimulated the enzyme to increase their number of end products. And if the number of end products are produced more than the expectation, then also this end products, they with the high concentration of end products, again they stimulates the enzyme to lower its activity. So anyhow, this end products, they regulate themselves. They regulate themselves by stimulating the enzyme. That's why this allosteric inhibitors are called as feedback mechanism. So if they are produced less than the expectation, then they with less concentration effect, they stimulate the enzyme. And if the molecules are produced more than the expected number, then also they regulates or influences the enzyme. Then also they stimulates the enzyme to lowers its activity and decreasing the end products. That's why it is called as feedback mechanism. So here product is regulating itself or regulating its own. That's why this is called as feedback mechanism. So here allosteric inhibitors, they plays the role of this type. So here again, once again, we'll repeat this inhibitors. Inhibitors are the fourth factor for this enzyme activity, and usually inhibitors, they decreases the rate of a reaction, or they decreases the enzyme activity. And these inhibitors are usually chemical compounds. And under these inhibitors, we can discuss 3 types: competitive inhibitors, and non-competitive inhibitors, and allosteric inhibitors. Competitive inhibitors are the inhibitors which decreases the production rate gradually. That means substrate molecules and inhibitor molecules both equally shares the active sites of the enzyme and gives the complexes which are temporary. But here, this enzyme inhibitor complex, it is not temporary, it is permanent. So this temporary enzyme substrate complex furtherly produces or dissociates into product and enzyme. So in this way, this 50% again gradually that decreases in giving the end product. So in this competitive inhibitors, they occupies 50% of the active sites of the enzyme. Then for this examples are one is succinate, where malonate acts as an inhibitor, and second is PABA, where this sulfur drugs plays the role of inhibitors. So then competitive inhibitors or competitive inhibition, it is a reversible mechanism. It is a reversible mechanism. Why? Because substrate molecules concentration that helps in increasing the microbes. That's why it is reversible. Then next, non-competitive inhibitors. This non-competitive inhibitors usually this non-competitive inhibitors gradually, that means substrate molecules and inhibitors both are ready to occupy the active sites, but this inhibitors, they occupies the other than active sites of the enzyme. By the binding of these inhibitors with the enzyme, enzymes they undergo conformational change. That means they changes their shape along with the active sites. As a result of this conformational change, substrate molecules cannot come and fit in the active sites of the enzyme because of their change in shape. So as a result of this changes, no end products are, that means a complex enzyme substrate complex will not be framed, and which cannot dissociate into end products. As a result of this, a total reaction will be held and there is no formation of end products. And third one is, for this examples, cyanide acts as a cytochrome oxidase, and for this penicillin is a non-competitive inhibitors. And in allosteric type of inhibitors, there is a self-controlling mechanism. That means end products, they controls themselves by stimulating enzyme. That means if the expected molecules are not expected end product molecules are not synthesized, then this less concentration end product molecule stimulates the hexokinase enzyme and activates them to increase the rate. If the end product molecules are more than the expectation, then with this higher concentration end product molecules, they again regulates or stimulates the enzyme in lowering its activity in producing the end product. So in this way, in both the ways, this end products controls themselves. That's why it is called as feedback mechanism. And this is about the allosteric inhibitors. But this enzymes, the total enzymes chapter will be completed. See you in the next class.